and welcome to the ACR Journals on Air podcast. I'm your host, Vicky Shamagam. Over the last 30 years, many thousands of patients with rheumatic diseases have enrolled in clinical trials. But have you ever wondered what valuable treasures might be hiding in plain sight in the placebo arms of those trials? Well, our guest today did just that. The manuscript we will discuss today is Peripheral Blood Gene Expression Profiling and Prognostic Significance for the Course of Interstitial Lung Disease in Patients with Systemic Sclerosis. It was published in Arthritis and Rheumatology, and a link to the manuscript and disclosures can be found in our show notes from today. It's a real pleasure today to be joined by the first author of this manuscript, Dr. Shervin Assassi. Dr. Assassi is the Director of Rheumatology, Professor, and the Norana B. Warner Endowed Chair in the Division of Rheumatology at UT Health Houston. He graduated from the Albert Ludwig Medical School in Freiburg, Germany, and then completed his internal medicine training and rheumatology fellowship at UT Health Houston. He co-directs the UT Health Scleroderma program, which houses one of the largest specialized systemic sclerosis programs in the USA, and has importantly contributed to elucidating the genetic basis and molecular profile of this multifaceted disease. Dr. Assassi is the wonderful rheumatologist, a caring physician, and a great friend, and it's truly a pleasure to have him join us on the show today to share his work on how to find hidden gems in the placebo arms of clinical trials. Welcome to the show, Shervin.
Hello, I'm delighted to participate in this podcast.
So could you begin, Shervin, by orienting us to the context of this particular study? What were the questions you were trying to answer?
Interstitial lung disease, abbreviated ILD, is the primary cause of disease-related death in systemic sclerosis. Based on the results of the NIH-funded Scleroderma Lung Study 2, mycophenolate has become the most commonly used medication for treatment of SSc-ILD. However, around 1/3 of the patients continue experiencing lung volume loss despite treatment with mycophenolate. So there is a heterogeneity in response to mycophenolate. And it is pertinent for clinical care and clinical trial design to identify those patients who will not respond to mycophenolate. However, no clinical or laboratory markers can reliably predict response to mycophenolate. So there is an unmet clinical need for predictive biomarkers. In a previous study conducted in Scleroderma Lung Study 2, in close collaboration with investigators at UCLA, we have shown that the peripheral blood gene expression profiling might be helpful in predicting who will respond to mycophenolate. In that study, we showed that high lymphoid lineage predicted better ILD course when patients are treated with mycophenolate, and high myeloid lineage predicted worse ILD course when patients are treated with mycophenolate. However, in that clinical trial, the comparator was not a true placebo arm. The comparator was active treatment with cyclophosphamide. So there is no true placebo arm in that trial. Therefore, we don't know whether these gene expression modules are predictive because of natural history of disease, or they're predictive because they're identifying patients who are more likely to respond to mycophenolate. Hence, we asked whether we can use the valuable samples and data from SENSCIS trial to replicate our results in Scleroderma Lung Study 2.
Thanks for that background and explanation. Tell us a bit more about the SENSCIS trial. It stands for the Safety and Efficacy of Nintedanib in Systemic Sclerosis. Tell us about that trial. Why were the samples collected in that study unique, and why did they give you this opportunity to do this type of a secondary analysis to look at gene expression profiles to see what might predict response to mycophenolate?
SENSCIS was a very large, actually the largest SSC ILD trial ever conducted. It's an international trial which increases the generalizability of any results based on that trial. And it investigated the safety and efficacy of an antifibrotic agent called nintedanib in comparison to placebo. The primary outcome of this clinical trial was met, and it showed that nintedanib slowed the decline in forced vital capacity, which led to the FDA approval of nintedanib for treatment of SSc-ILD. The large sample size, as well as the standardized treatment regimens and standardized data collection in this trial, represent a unique opportunity for biomarker development or validation.
So these analyses were not part of the original SENSCIS trial plans. How did you, as a scleroderma specialist, but not someone who was working directly for the sponsor of the trial, how did you persuade the SENSCIS trial organizers to let you have access to the samples and the data?
These type of collaborative efforts require mutual collaboration as well as long-standing working relationship. I'm grateful to trial leadership and scientists at Boehringer Ingelheim for working with me on publishing these results. This study exemplifies nicely the importance of academia and industry collaboration and partnership for biomarker development studies. Contrary to observational cohorts, patients are treated according to standardized regimens, and outcome measures are collected at fixed time points in clinical trials. Hence, they are really a treasure for biomarker development and validation. I worked early on with scientists at Boehringer Ingelheim to actually design the protocol for sample collection. And then once the study was concluded, I worked with them closely to analyze the data, and they were open to my ideas and open to validating study results of another clinical trial that was actually funded by the NIH. So it requires really a partnership between academia and industry.
Yeah, I totally agree. And tell us a little bit more about the patients who were enrolled in SENSCIS. Why, and I guess you kind of told us a bit about this already, why were you so focused on the placebo arm of this particular analysis?
So all patients in SENSCIS trial had to have ILD. Specifically, they had to have more than 10% fibrotic changes on high resolution chest CT. What is so special about SENSCIS trial, contrary to other previous SSc-ILD trials, SENSCIS actually permitted stable background MMF treatment. The patients that were on an MMF had to be on stable dose MMF for 6 months. And among 288 patients assigned to the placebo arm of SENSCIS trial, around half of them were on stable dose mycophenolate, and the other half, the vast majority of the other half, was on no DMARD. More than 90% of the other half was on no DMARDs. So half of the placebo is on stable dose mycophenolate, the other half is on no DMARD. So that represents a really unique opportunity to examine A, what happens when patients are on stable dose mycophenolate treatment, B, what happens when we are investigating the natural history of disease.
Yeah, it's amazing study data, huge number of patients, and really insightful that you were able to think to do this. I'm super impressed. So tell us a bit more about the mechanics of the study itself. How did the peripheral blood samples get collected and analyzed?
So the peripheral blood samples were collected in a collection system called PAX gene tubes. This collection system stabilizes RNA up to 5 days at room temperature, which is really great for sample collection in multi-center international clinical trials. And it avoids storage and handling artifacts resulting from instability in RNA. So it's convenient to collect, it's convenient to transport, and it's really robust to artifacts resulting from storage or handling. So I was very fortunate that whole blood RNA was collected in the SENSCIS trial.
Was it analyzed at your center or by the study centers?
The gene expression data were generated by Boehringer Ingelheim, but the analysis was conducted jointly. It really followed the same pattern, and I was very grateful to the Boehringer Ingelheim scientists to do that, that we used in the Scleroderma lung study too. Basically, the goal was to replicate those results.
Mhm.
In order to replicate those results, we had, for example, to use the same list of genes that were used to define modules in Scleroderma lung study 2.
Yeah, that makes sense. And what measures did you use to assess progression of lung disease?
The primary analysis was based on absolute change from baseline in forced vital capacity percent predicted at week 52, which basically at 1 year. But we also did secondary analyses where we dichotomized the change in FVC percent predicted based on a cutoff of 3% for worsening or improvement, which has been found to be the MCID for FVC percent predicted in SSC-ILD based on a previously published study.
Yeah, and then tell us a bit more about this modular analysis approach and how you used the peripheral gene data to assess predictive significance. Can you walk us through that analysis?
Basically, each time we perform gene expression profiling, we are getting the results, long list of genes. Often we have not even heard the names of those genes. So we are really in that data indigestion phase. There are methods for data reduction in a scientifically meaningful, and the modular analysis developed by Chassabel et al. That has been applied to many autoimmune diseases is one of those data reduction methods. It's based on list of genes that are co-expressed across many human diseases, such as autoimmune diseases, infections. So these genes go together, and then we can calculate a modular score for all those genes, all those co-expressed genes. What they have also done is they have gone back and assigned biological functions to those genes that are go together, that are co-expressed. For example, there's a module for lymphoid lineage, there's a module for myeloid lineage. Instead of looking at thousands of genes, we are looking at 62 curated whole blood modules. That is much easier to understand, but also much easier to handle statistically.
Yeah, that makes sense. And there is a visual of this in your manuscript in figure 1, where you show the associations between the baseline annotated modular scores and the change in FVC. Can you describe that for our listeners and explain to us what it shows?
Sure. That figure shows two volcano plots. One panel is for the patients who are on stable dose MMF. The other panel is for patients who are not treated with MMF. MMF stands for mycophenolate. And within each volcano plot, the right side modules are associated with improved FVC, and the left side modules are associated with worsened FVC at 52 weeks.
In the SLS2 patients who were taking mycophenolate, you had seen higher baseline levels of lymphoid lineage and mitochondrial or protein synthesis modules associated with a slower decline in FVC. Did you see similar findings in this data set?
The answer is yes. And I was personally surprised how much overlap there was between the results in the SENSCIS trial and Scleroderma lung study 2. Because obviously these were very different trials, different study population, different inclusion and exclusion criteria. And in the Scleroderma lung study 2, patients were on no immunosuppressive agents and were started on mycophenolate, whereas in SENSCIS, they were on stable dose mycophenolate. But the same modules that predicted better course of ILD in Scleroderma lung study 2 also predicted better course of ILD in SENSCIS trial when patients were treated with mycophenolate. Basically, higher baseline levels of lymphoid lineage, mitochondrial and protein synthesis modules were associated with better course of ILD at 52 weeks in both trials.
Really looking at that predictive significance of the modular gene expression scores for FVC worsening or improvement, the finding was the same. The myeloid modules were increased. Am I understanding that correctly?
Yes, so basically there are some modules that are associated with better course. There are some modules that are associated with worse course when patients are treated with mycophenolate. We also had obviously the luxury in SENSCIS trial to have also half of the patients not being treated with mycophenolate. Basically, in half of the placebo arm, we were looking at a natural history of disease. That's a golden opportunity to differentiate prognostic versus predictive biomarkers. Prognostic biomarkers prognosticate the course of disease regardless of treatment, regardless of whether you are treating the patients with mycophenolate or not. Whereas predictive biomarkers are biomarkers that predict response to a specific treatment. What we could show, capitalizing on really this valuable sample and clinical data collection, is that patients that are treated with mycophenolate do better if they have a higher baseline lymphoid protein or mitochondrial modular scores, and they do worse if they have a high myeloid signature. When we looked at the other half that was not treated with mycophenolate, there was only a signal on the myeloid signature. On that signature, patients who had a high myeloid signature did worse at 52 weeks, regardless of whether they were treated with mycophenolate or not treated. That's a prime example for a prognostic biomarker. So treatment really doesn't matter. But when we looked at those modules that were associated with better course, those modules only panned out in the half that was treated with mycophenolate. So those modules are a prime example for a predictive biomarkers, biomarkers that have a prognostic value only if the treatment is rendered. And that is also consistent with how mycophenolate works. Mycophenolate goes after activated lymphocytes. It makes sense when patients have a high lymphoid module that they respond better to mycophenolate. But mycophenolate typically doesn't go after myeloid cells. So it also makes sense that patients do worse if they have a high baseline myeloid signature.
Yeah, that's really helpful information. I'm going to ask you another question about some of the analysis you did and then come back to what you just said. So during the analysis, you also studied the correlation between C-reactive protein, baseline C-reactive protein, and the baseline gene expression. Was that analysis insightful? Tell us what you found.
That analysis was conducted because we have previously shown, both in observational cohorts as well as in Scleroderma lung study 2, that patients who had high CRP had a worse ILD course. And we wanted to know whether this myeloid signature is just a surrogate for CRP, which is a general inflammatory marker. Basically, are we doing a fancy CRP?
Very expensive CRP.
Exactly. Then it wouldn't be meaningful. Then we could just order CRP. And what we could show that there is some correlation between the myeloid signature and CRP. Otherwise, the myeloid signature would not have face value. But the correlation is really weak. The correlation is zero point 0.2, the correlation coefficient, which means the myeloid gene expression signature and the serum CRP do not provide overlapping information.
So, in this study, some of the patients had started mycophenolate, as you said, at least 6 months before enrollment. So, some of those baseline samples, your patients were receiving MMF. We've talked a little bit about the baseline lymphoid and mitochondrial protein synthesis modules. I'm wondering, do you think this data is something that might eventually be useful in clinical decision making?
I believe these data can be helpful in clinical decision making because around half of the patients with SSC-ILD that present to tertiary referral centers are already treated with mycophenolate. That has become our new normal. Mycophenolate has become for SSC-ILD is methotrexate for RA. The threshold for starting mycophenolate is low. Then we have to know, okay, these patients are on stable dose mycophenolate. Can we predict who is gonna have a worse ILD course despite being treated with mycophenolate? These results provide some answers.
And it sounds like these results tell you that you can predict it more effectively with this gene expression analysis than just with CRP alone. Am I understanding correctly?
I do believe CRP has a role, but our study indicates that CRP and these gene expression modules don't just simply provide overlapping information. We have previously shown that if you have a high CRP and are treated with MMF, you have also a worse course. So I think both are helpful.
That's really interesting. So tell us a little bit about the limitations of this data. Why should we still be cautious here?
Sure. So the strength is now we have validated the results of Scleroderma lung study 2 in a totally independent patient sample with different inclusion exclusion criteria, with a geographically different catchment area. It increases the generalizability of the results. However, the predictive biomarkers identified in this study cannot, for example, be applied to other immunosuppressive agents that are also being used for SSC-ILD, such as rituximab or tocilizumab. So I would love to do similar studies in clinical trials that investigated the efficacy of those two drugs. And what is also important to point out is that these patients were on stable dose mycophenolate for 6 months. But we didn't investigate when mycophenolate was started.
Hmm.
Was it just 6 months ago, or 1 year ago, or 2 years ago? So there is some variability there. So a study that would examine that in a systematic fashion would be very helpful.
It's all great questions that as a clinician you really want to know the answer to. So studies studying outcomes in scleroderma lung disease are notoriously difficult. Do you see a role for this type of peripheral gene expression profiling at enrollment into clinical studies where it could be used potentially to stratify enrollment into the different arms?
I do believe these biomarkers might play a role in stratifying the patients because most SSC-ILD studies now permit stable dose mycophenolate background treatment because it would be unethical to have true placebo arm now. So we want to enroll patients into SSCID trials that have progressive disease despite mycophenolate treatment. Otherwise, we are going to end up with just negative clinical trials. If we enroll patients who are just stable and
Mhm.
don't have progressive ILD when they are on background mycophenolate treatment, I believe blood gene expression profiling might be helpful in that setting.
I agree. I think it's really fascinating and maybe could accelerate some discoveries in that area. So, Shervin, that was a really wonderful dive into the very deep world of scleroderma lung disease. I wondered if we could take a slightly different direction in our conversation now and take a dive into your career. You grew up in Iran. You later moved to Germany, where you attended medical school. How did you land in Texas? And what has made you stay on in the department where you trained?
So I knew already during my medical school training in Germany that I want to pursue a career in rheumatology. I found rheumatology fascinating. I loved immunology. I loved anything that has to do with joints. And rheumatology was the perfect combination for me. And that's why I was focused on universities that have a strong rheumatology program, as I was interviewing actually for my internal medicine residency program. When I came for an interview to UT Health Houston, I was not only impressed by the strength of rheumatology program at UT Health Houston. I interviewed at that time with Dr. Arnett, but I was actually blown away by the enormous resources and infrastructure at the Texas Medical Center. I'd never seen such a thing where several top notch medical institutions like MD Anderson, Baylor College of Medicine, and Texas Children's Hospitals are located within walking distance of each other. And last but not least, I found Houston to be a wonderful city to live in. It is very welcoming. As an immigrant, I feel you pack your backpack and you're on the road and you put down your backpack at some point in a city where you feel welcome, but also you feel you could build a life there. And that was Houston for me.
Yeah, it's amazing. And you now lead that division. Running a division of rheumatology is very hard work. Tell us about your experience as division director. What do you wish you'd known before you took on the role? And do you have any tips in juggling the many, many tasks that every day brings?
Sure, being a division director is hard work, but it's also a lot of fun. For me, being a division director at UT Health Houston is especially a privilege. I have been in this division for more than 25 years. I have been in this division longer than I have lived in my mother's house. So I have been very fortunate to work in an environment that is supportive and encouraging. This environment was not created by me, but was created by the university and divisional leadership long before I became a division director. And my goal is to continue that path. As a division director, working with my faculty, I can identify growth areas and set goals and really work together to materialize them. Each member of our team is at a different career stage, has different goals, and I see my job giving them the necessary resources and empowering them to reach the goals that they have set for themselves. The part that I wish I knew before I became a division director is that as a division director, you have to be ready for unplanned events,
Mhm.
sometimes positive, sometimes negative. I became division director 2 months before the COVID 19 pandemic. So you can imagine how much surprises I would encounter on daily basis. So understanding that part of the job would have made my life easier. Basically, embracing the fact that your day might not go as planned and being flexible to do changes to your daily schedule to address issues before they become problems.
Yeah, it's a very good advice. And predictability of unpredictability is definitely a truth of roles such as yours. So you have managed to stay very focused on your research despite all of these competing priorities. You've remained extremely productive publishing your work. I'm wondering, would you share any tips on how do you like to write? Are you someone who sets aside specific writing time, or do you chip away at perfection in the many pieces of time confetti that make up your day?
I have stayed focused because I really enjoy analyzing data, interpreting collaborative analysis results, and writing about them. As you know, writing is a skill that one learns over time. One gets faster with practice, and one develops a pattern for writing scientific manuscripts over time that fits to one's strengths. Having said this, I firmly believe that one shouldn't multitask. One needs dedicated time for deep work, and writing counts as deep work because otherwise one becomes inefficient and the quality of the end product suffers. So I do set aside time for writing. I love working early mornings. I have been like this since I was a student in the school. And sometimes I also write on the weekends because that is the time I have where I can focus on writing uninterrupted.
A follow up question to that, Shervin, and I appreciate you sharing the importance of focus, is sometimes it's not just what a person chooses to spend time on that makes them successful, but what they choose to not spend time on. So, do you have any pointers on how you decide what you're going to say no to, and indeed on how to say no?
Sure, prioritizing is not an easy task. I'm still working on it. It's really an ongoing process. It requires knowing your own priorities. You have to really ask yourself, what is your goal? For me, the goal has been doing meaningful research that can improve the lives of people living with systemic sclerosis or fibrosing interstitial lung disease. That is the primary focus of my research. So, activities and tasks, especially in the research area, that are not focused on that goal, unless it's mentoring other juniors in other diseases, which is totally different, are distractions and are not helping me to reach my primary goal. So I regularly make lists between my goals and activities to identify mismatches. So I spend time actually making lists. And at this point, if I say yes to a new activity, I ask myself, have I already completed a previous activity or willing to delegate to another person an ongoing activity? Which brings me to the most important point about time management. Successful research or administrative operations are not one man show. They require teams, consisting of motivated and competent scientists, administrators, etc. So spending time on team building is actually a part of time management and is a priority for me.
Yeah, that's really good advice. So, as part of that, what habits do you make time for even when you're busy, stressed out, overwhelmed, trying to make sure that everybody else is hitting their RVU targets and meeting their own goals? What keeps you anchored and lets you stay consistent?
So spending time with my family is a great way of recharging and getting out of the work mode. Simple things like eating dinner together as a family, where all family members come together after a busy work day, or having deep, meaningful conversations can create a balance to busy work life. Having hobbies, if time permits, outside the work can also help to reset energy. When time permits, I jog, swim. That's a good way to recharge. I also listen to one book per month while driving, which I greatly enjoy.
Yeah, I'm a huge fan of audiobooks myself, so I share that habit for sure. You are very fortunate that you've had some outstanding mentors throughout your career. I'm wondering, would you share with us some of the mentors who've touched your life? What did you learn from them? And what do you try to pass forward as you're now mentoring the next generation of rheumatologists?
So I have been very fortunate to have had outstanding and supportive mentors throughout my career. Dr. Mays, Dr. Tan, and Dr. Arnett were my mentors in the field of systemic sclerosis. They have taught me that one has to work with other centers, with other researchers across the nation and actually across the world to conduct meaningful research in a relatively rare disease such as systemic sclerosis. They have taught me the importance of embracing new technology, new methodology. One cannot just keep doing the same thing over and over again. Research moves fast. What is innovative today becomes outdated tomorrow. I have also had methodological and career mentors outside my division, and I would actually highly encourage others to have mentors outside their division. My mentor was Dr. John Tyson, who has helped me to stay focused and give me an outside perspective. Lastly, my former division director, Dr. Ravel, has enabled me to make a career in a positive and supporting work environment, which I try to continue for my faculty and mentees now. Also, my mentors have taught me it's not only important to have a mentor that can guide you in answering scientific question, your mentor has to be also your sponsor, has to help you with networking, has to plug you in into multi-center collaborations. And I try to do the same thing for my mentees.
Yeah, I think that's a really, really good advice. I'm going to throw you a curveball now because you told me right before we started recording today that you actually had early on at the inception of this podcast kind of been involved in the decision for there to even be an ACR Journals on Air podcast. Now, as a guest on the podcast, I'm wondering if you could reflect back on that decision and tell us why the Committee on Journals had decided to have a podcast.
I was chairing the Committee on Journal Publications when this idea was brought up. It was felt nowadays our colleagues are accessing information differently, and a podcast would be very helpful to showcase the amazing research that our ACR journals are publishing. I have to say, I am so impressed what you and other colleagues have done with this podcast. I greatly enjoy listening to it, and I am delighted to be on this podcast as a guest.
Oh, Shervin, thank you. It's really nice to hear, and so glad that you came up with the idea and you and your colleagues voted this through. I want to thank you, Shervin, for such a great discussion today. Thank you for navigating the complexities of this particular study. Thank you for explaining it and sharing your insights with us today. It's always a pleasure to talk with you, and I'm really excited that you were able to join us and share all your expertise with our listeners.
Thank you so much for having me.
So I want to close today's episode also with a special thank you to everyone who decides to enroll in a clinical trial. The gift you give is profound. Your time, your data, your lived experience, which become the foundation for discoveries that will help people you will never meet. But by allowing researchers to learn from your journey, you're truly paying it forward to future patients who will face these rare diseases with more options and certainly more hope because of you. I also want to thank the study sponsors who not only design trials that answer primary clinical questions, but who through their generosity can also make it possible to conduct secondary analyses, ensuring that valuable data that might otherwise remain unseen can be used to address pressing scientific questions and drive this field forward. Thank you all for listening to this podcast. ACR Journals on Air is a production of the American College of Rheumatology. Please follow at AC Room on social media, email us at podcast@rheumatology.org, and rate and subscribe to our podcast at acrjournalsonair.org or wherever you get your podcasts.
This podcast was edited by Resonate Recordings.